Pharmapal.
Welcome to our YouTube video on lead generation and lead optimization in drug discovery. In this video, we will explore the process of finding the best chemical compound that has the potential to become a successful drug. So, let's dive in. The goal of a lead optimization program is to identify a chemical compound that, after extensive testing, has the highest chance of becoming a successful drug. But before we can optimize a lead, we need to generate one. Lead generation is the initial phase where we start the search for potential compounds. We have two choices: small molecules or biologics. Small molecules are typically produced through chemical synthesis, have a molecular weight below 500 Da, and can be taken orally. On the other hand, biologics have a higher molecular weight, are usually delivered through injections, and are more expensive to manufacture. Historically, most successful drugs have been small molecule organic compounds. Some examples include Lipitor, Abilify, Advair, and Plavix. However, biologics have been gaining popularity. Best-selling biologics include Enbrel, Humira, Remicade, and Avastin. When optimizing a lead, there are several desired features we aim for. These include efficacy against the disease, minimal side effects, and the ability to be taken orally. We also want the drug to be chemically stable, have a reasonable drug dosage, and be patentable to ensure exclusivity. Now that we know what we want, let's talk about the lead generation phase. This is where we choose a compound as a lead, which is a major judgment call. The lead should have high potency, high selectivity, and good pharmacokinetics. One approach to lead generation is natural product screening. This involves searching for potential drug leads in natural sources, such as plants or bacteria. While this approach has been successful in the past, it can be resource-intensive and may not be suitable for all therapeutic areas. Another approach is high-throughput screening, which is the main source of drug leads in modern drug discovery. This involves testing thousands of compounds per day using biological assays. Major drug companies have extensive compound libraries for this purpose. If high-throughput screening is unsuccessful or unavailable, fragment-based lead discovery can be pursued. This approach relies on the idea that drug molecules are made up of individual binding fragments. It involves biophysical screening methods and the optimization of initial fragments. Once an initial hit is identified, it may undergo a limited optimization process before committing resources to a full lead optimization program. This involves improving the compound's profile, such as its potency and pharmacokinetics. The lead optimization program involves scientists from various disciplines, including chemistry, biology, and drug metabolism. It typically lasts 2 to 3 years and may involve synthesizing and testing several thousand compounds. At the core of a lead optimization program is a screening paradigm, which outlines the major biological and drug metabolism assays used for compound selection. This multidirectional and iterative process helps guide the optimization efforts. One important aspect of lead optimization is target affinity optimization. This can be done through empirical approaches, such as homolog synthesis, isomer synthesis, or bioisosteric replacements. It can also involve rational approaches, such as using protein structures to design virtual compounds. Another crucial aspect is optimizing the absorption, distribution, metabolism, and excretion (ADME) profile of the compound. This helps determine its systemic exposure and overall efficacy. Strategies for optimization include reducing metabolism, prodrugs, and soft drugs. Minimizing CYP450 inhibition is also important to prevent drug-drug interactions. Inhibition of CYP450 enzymes can affect the metabolism of co-administered drugs. This is a key consideration in lead optimization. To illustrate the lead optimization process, let's look at a case history. The identification of selective 5-HT2C receptor antagonists for the treatment of depression and anxiety. The 5-HT2C receptor is involved in anxiety and depression. Selective antagonists can potentially treat these disorders. The lead compound was a competitor's compound with reported selectivity. The lead compound had some limitations, including insolubility. Conformationally restricted analogs and bioisosteric replacements were explored to improve potency and selectivity. Docking studies were performed to understand the interaction of the lead compound with the receptor. This helped guide the design of new analogs with improved binding affinity. After several rounds of optimization, SB243213 was identified as a potential development candidate. It showed good activity in animal models with no side effects and improved solubility. In summary, lead generation and lead optimization are crucial steps in the drug discovery process. Through various approaches, scientists aim to find the best compound with the desired features for a successful drug. Thank you for watching our YouTube video on lead generation and lead optimization in drug discovery. We hope you found it informative. If you have any questions, please leave them in the comments below. Don't forget to like and subscribe for more content like this. Until next time.
Pharma Pal.